Hello, hi, my name is Hideho Okada at UCSF. I thank the organizers for this invitation, and I'm going to discuss immune modulation and immunotherapy for DMG and DIPG. Here's my relevant disclosure information. And there have been a few excellent publications reporting the immune microenvironment of DIPG and DMG in comparison to adult GBM. A collaborative study between Harvard and Stanford group demonstrated that DIPG cells produce low cytokines and chemokines associated with fewer T cells and less inflammatory myeloid cells. Here, upper right panel, the vast majority of CD45 positive hematopoietic cells in the DIPG are CD11B positive myeloid cells, just like you see in normal brain cortex, while adult GBMs contain non-myeloid populations such as T lymphocytes. Moreover, as you see in the right lower volcano plot, the DIPG infiltrating myeloid cells produce no chemokines or cytokines as opposed to those infiltrating adult GBM. And a group from Seattle children also reported similar findings, such as the lack of PD-L1 expression, which is normally induced by interferon gamma. A group at the University of Cincinnati recently reported using both PDX and in utero electroporation mediated mouse models of hemispheric HGG high-grade glioma and DMG models that in DMG, the blood-brain barrier is quite intact, while the vessels are enlarged and become quite leaky in high-grade gliomas. The TMA dextran indicates the vascular permeability, and the TER119 indicates the extravasation of red blood cells, which was seen in high-grade glioma model, but not in DIPG. As you can see, the characteristics of DIPG are almost identical that you see from in normal control brain. In terms of T lymphocyte homing across the blood-brain barrier, as reviewed by doctors Marchetti and Engelhardt, without inflammatory status, which was on the left, activated T lymphocytes can still transmigrate through the initial endothelial basement membrane, get to the subarachnoid CSF space. However, they are not able to cross the glia limitans, which is the second layer of the BBB. That's why in most cases, even if you see T cells in the brain or brain tumors, those T cells are mostly stuck in the perivascular space. On the other hand, under the inflammatory conditions, especially when antigen-presenting cells can present cognate antigens to the T cells, those activated T cells can now transmigrate through the glia limitans by molecules such as matrix metalloproteinase, MPP2 and MPP9 as a key mediators. It is also important in the initial process that the T cells need to be sort of arrested by the interaction between the adhesion molecules on the endothelial cells, such as VCAM1 and ICAM1, and corresponding receptors such as LFA1 and VLA4 on T cell size. So my laboratory has been studying about critical factors that can mediate effective T cell homing to the brain tumor site and the pharmacological intervention to facilitate the process. As you can see here, this is a brain infiltrating vaccine reactive T cells in this mouse model. as you can see that if you combine vaccine and the poly-ICLC, which is a ligand for toll-like receptor 3 and MDA5 double-strand RNA mimic, you see a synergistic enhancement of vaccine reactive T cell infiltration to the brain compared to poly-ICLC only or vaccine only. And we found that these T cells express high levels of brain homing receptors such as CXCR3 and VDA4. So we reproducibly shown and also in other publications that poly-ICLC can enhance these receptor expression in the brain and also actually ligand expression in the brain tumor sites such as CXCR10. So that combination of the vaccine poly-ICLC induced remarkable and almost synergistic anti-tumor effects. So through Pacific Pediatric Neuro-Oncology Consortium, PNOC, we developed and conducted a multi-center phase 1 vaccine trial targeting histone variant 3 K27M mutation. So we enrolled and treated a total 29 DMG patients with the K27M peptide and poly-ICLC. We developed a novel mass cytometry-based evaluation of the vaccine reactive T cells in depth, not only the numbers, but precise phenotype and activation exhaustion status of those T cells. We also evaluated a role of myeloid cells in the peripheral blood. Then we found that overall survival and progression-free survival are actually associated with the induction of vaccine-specific effector memory. Let me go a little detail in the next slide. So in this study, we enrolled newly diagnosed 19 DIPG patients and 10 non-DIPG DMG patients. Following the initial diagnosis of the K27M H3.3 K27M tumor by biopsy and also confirmation of HLA 0201 positive status, patients undergo standard of care radiation therapy, followed by a combination of peptide vaccine targeting the mutation and tetanus toxoid as a helper in combination with poly-ICLC for every 3 weeks. And we monitor peripheral blood T cell response and myeloid cell response. In clinical trials of cancer vaccines in general, immunological response data are not always associated with clinical outcomes. However, in our study, we found that expansion of vaccine antigen-specific CD8 cells, especially effector memory cells, associated with patients' overall survival and progression-free survival. On the other hand, if you look at the expansion of vaccine reactive based on mass cytometry, but if their phenotype is associated with more like an exhausted status, the expansion of those exhausted cells was not associated with either overall survival or progression-free survival. In our publication, we also reported the effects of dexamethasone treatment on the vaccine response and the possible roles of myeloid-derived suppressor cells as a biomarker. So please refer to our publication for more information. So in summary, the tumor microenvironment of DMG and DIPG is characterized with fewer T cells and less inflammatory myeloid cells compared with adult GBM. The blood-brain barrier of the DMG DIPG appears to be intact, just like normal brain. The better understanding of the T cell homing through the BBB should help us to formulate improved strategies. The PNOC 007 vaccine trial using peptide and poly-ICLC demonstrated the expansion of the antigen-reactive CD8 positive effector memory T cells, but not exhausted T cells, is associated with better overall survival and progression-free survival. Though we noted that the limitation of the study was that we didn't get many tumor tissue, post-treatment tumor tissue to evaluate directly the impact of vaccine treatment on T cells. So this study was contributed by many of my laboratory members and PNOC consortium members and site investigators, Parker Institute for Cancer Immunotherapy, UCSF Neuro-Oncology Biostatistics, and core services, the UCSF Cancer Immunotherapy Lab, industries and fundings, and most importantly, patients and their families. Thank you so much.